Good afternoon, students. Myself, Mrs. Ashwini Kadbhane from the Department of Microbiology, S.V.K.T College, Tivlali Camp. Today we are going to see the TY-BSc Microbiology paper number 3, that is the Enzymology. In that we are going to see the unit number 1, that is the purification of the enzymes. In that we are going to see purification of enzymes based on the molecular size. In molecular size, the two methods they are generally used. One, it is the gel filtration method, and the next one, it is the density gradient method. Today in this session we are going to see about the gel filtration method. How the gel filtration method is, what is the principle behind the gel filtration, how enzymes are purified by using the beads or using the matrix or by using this technique, and what are the limitations, advantages and the disadvantages of this technique. Before starting my lecture, I am going to revise what we have learned earlier. Earlier we have learned the purification method based on the specific binding. In that we have seen about the affinity chromatography, we have seen its basic principle, we have seen what is ligand in that is one specific like enzyme substrate or antibody antigen. This method is very specific. So we have seen its procedure, we have seen its advantages, limitation and the disadvantages and its applications. So in our today's session, we are going to learn the principle based on the molecular size of a particle, that is the molecule which we have to purify, that is the enzymes or the proteins based on its molecular size. Okay, we are going to say its principle, its components used in this technique, its applications, its advantages and the disadvantages. So before that, again we are going to see what is purification of enzymes. Any functional study of enzymes cannot be performed until we obtain their homogeneous preparation. Okay, so when we said a crude means mixture. All the molecules, they are mixed in that particular samples. That is the crude sample. From the crude sample, we have to prepare a homogeneous mixture, that is a purification technique. And for the purification technique, there are various aspects on the molecular size or on the specific binding capability or on the ion capability or on the solubility differences. So here we are going to see about the molecular size. Homogeneity of enzymes, that is very essential for the application like the therapeutic use. Enzymes being proteins, they are separated on the basis of their solubility, charge and the size. Cell is broken to prepare the cellular extract or the crude extract, bringing enzyme in the solution, which is then separated to the other proteins on the basis of their physical properties like the size, charge and its solubility. So protein purification method, before purifying any type of protein or the enzymes, one should have focus on some points. Okay, when you are purifying any molecule or any enzymes, these things should be focused, because whatever you are purifying, it should have the high recovery rate. If you are purifying an enzyme from the crude, mostly 90% enzyme should be recovered. Then and then only it can be economically good. If we can't purify the enzyme on the high rate or we can't recover the enzyme on the high rate, it would be very cost effective method. So the point should be focused while purifying any enzymes or the proteins. They are the high recovery rate, highly purified enzyme protein, reproducibility of the method should be there, economically use of chemical should be there. When we are using any method, that method should be economical and it should have a shorter period of time for the completion of the method. If the time period is more, then it is very difficult for a factory or industry to purify that particular enzyme or the particular protein, because it would be economically high. So before starting, we are going to see about the column chromatography. So this basically it is the type of a column chromatography. Affinity chromatography is also type of a column chromatography, where a column is prepared. In that column, all the materials, in affinity we are using a ligand, we are in that stationary phase or the mobile phase we are using. Here also there are two types of phases. In each and every chromatography, there are two types of phases. that is the stationary phase and the mobile phase. In stationary phase we are using various gels, and in the mobile phase we are using the beads. Okay. So in column chromatography, if it is uses the property of size, it is called as gel chromatography. If we uses the charge, it is called as the ion exchange chromatography. If we uses the affinity, it is called as the specific binding property. So all these three techniques, they are comes under the column chromatography. All these column chromatography procedures use the buffered solution. Okay, the mobile phase and the porous solid material held in the column, that is the stationary phase. Here the mobile phase and stationary phase, they are used. In stationary phase we are using the porous solid material, and in the mobile phase we are using the buffer system. Now we are going to start the actual technique which we are going to see in our today's lecture. That is based on the size of the enzyme, on the molecular size of the enzyme. This technique is generally also known as the gel sieve exclusion type of chromatography. Okay. So there are two techniques on the size. One, it is gel filtration, and next is the density gradient. Gradient means according to the grades. Okay, the molecules, they are arranged. But that we are going to see in the next session. Today we are only going to focus on the gel filtration chromatography. So gel filtration chromatography is also known as gel permeation molecular sieving or the size exclusion chromatography. It is a technique in which separation of the molecules or the components is based on the differences of their molecular weight, which is the size, and one is the effective method to isolate and analyze the bio-macromolecular substances. The bio means the live, and the macromolecule, which are more, that is the proteins or the enzymes or the nucleic acids. Okay, so to separate these types of molecules, we are using the gel filtration technology or chromatography. In gel filtration, there are two phases, that is the stationary and the mobile phase. Stationary phase, it is the consist of the beads containing the pores that span a relatively narrow size. When the gel is packed into a column and percolated with a solvent system means here we are using a buffer, and that would be a mobile phase for this type of chromatography. When the gel is packed into a column and percolated with a solvent, it permits a large molecular weight components to move faster without the penetration of the porous, that is totally excluded. Now what is happening over here? In the next slide we are going to see in the diagrammatic manner. But before that, there are the molecules which are having a particular size, and for this chromatography we should have the known, the target enzyme or the purify enzyme. We should know, the size should be known to us. If the size is known, particular on the basis of the size, we can design the beads for that. Okay. So when we are designing a beads, the small molecules that would be entrapped in the beads, and the larger molecule which have the larger size than the beads, they will be excluded from the beads and they will directly excluded from your column system. Okay. So the entrapment of your enzymes into that beads would be having a particular purification method for this type of chromatography. Smaller molecules spend more time inside the beads because they are entrapped in that particular beads. Larger molecules, they were percolate faster at the faster rate, and they would be excluded from that particular column. Smaller molecules spend more time inside the beads and therefore they are eluted later, while the larger molecule, after the larger molecule of the mobile phase has passed through the column. Now what is the principle of the affinity chromatography? The stationary phase consists of a support medium in which a substrate, here also we are using a ligand, is bound covalently in such a way that the reactive groups of that is essential for the binding of the exposed. So here the molecules, they are going to be used. Okay. In this figure, as you are seeing in this figure includes the various this figure shows you the diagrammatic way of the gel filtration chromatography. Here you can see the three types of colors. One, it is pink, blue and red. Pink denotes a very small molecule. Blue denotes the large or medium size molecule, and the red one denotes very large molecule which can exclude at the faster rate from the beads. See here the column is seen in this. The smaller molecules, they have been entrapped in the beads. The medium sized molecule, they are only attached to the surface of the beads, and the larger molecules, they are excluded from that particular bead, and they have excluded at the faster rate before coming before the smaller and the medium particles. So very small molecules entered many pores of the gel, equilibrating between the gel and the moving buffer, and so they travel very slowly and elute later. The medium sized molecule enters some pores in the gel, equilibrating between the gel and the moving buffer, and the larger molecules, they enter the few pores in the gel, so they travel rapidly and they are eluted at the very early stage when we start this. Now what are the properties and nature of the gel? What should be when we are using such type of chromatography technique, the gel which we are going to use, because gel, it is very important in this particular type of chromatography, because when you are putting the beads and the molecules inside that column chromatography, there is the stage between a gel and your bead, and gel, it is the moving, that is the mobile phase, and your bead, it is the stationary phase. So your nature of the gel should be very important part in such type of chromatography. So your nature of the gel, it should be a chemically inert, it should be have a stability, it should be a mechanically stable gel, it has some ideal and homogeneous porous structure, wide pore size results in very low resolutions. Okay, it should have a uniform particle and the pore size. The pore size of the gel must be carefully controlled, that is the degree of the cross linking. So these are some properties or the nature of the gel. The next thing, we are going to use the types of gel. Which type of gel you can use for such type of chromatography? First, it is the Cephadax gel, that is the dextrane type of gel. This is the 1,6 polymer of glucose which is prepared by fermentation of the sucrose sugar, that is the glucose and fructose. You know that sucrose sugar, it is generally and naturally present in the sugarcane juice. Okay, so this gel or this dextrane gel, it is prepared by fermenting the sucrose type of sugar, which is a carbohydrate, and it is a natural type of gel. Second one, you can use agarose gel, which is also a natural type of gel, which is obtained from the agar and composed of alternating units of 1,3 linked beta D galactose and 1,4 linked 3 N-hydro alpha L galactose. So it is also a type of a natural gel which can be used and which is called as the agarose. And third one, it is the acrylamide gel, which is a synthetic type of gel. It is not a dextran polymer, it is polymerized acrylamide or the methylene bis acrylamide gel. So it is also a synthetic type of gel. Now we are going to see the procedure, how we can perform or how we can go for the gel filtration chromatography, separation technique. Okay, so before proceeding towards this, we should have a column. How to prepare a column for gel filtration? A swelling of the gel is there. So swelling, that is the resin. We are using a resin. Resin we are using here, that is the beads. So swelling of the gel, some resins come in the powder form. They must be sonicated first in the elute and or on the desired buffer to swell. Okay. Because if there is no swelling of a resin, how the entrapment of your enzymes would be there in that particular beads or the particular resins. So swelling of the gel is very important over here. Then the packing of the column. You should have a slurry of gel plus buffer and pour into the column, which is 1/3 filled with the buffer. Washing the resin. After packing, pass several column volumes of the buffer through the column to remove any air bubbles and to test the column homogeneity. This was the preparation of column. Now we have prepared a column which is having the swelling of gel, which is having the packing of the column, and which is washing the resin. Now we have to load the sample. While loading the sample on the column, the sample must enter in the resin in the form of solution using a syringe. And last, it is the elution method. We have to elute the sample, and detection of components, the fractions are collected in the sample of the elutes from the column. So you can see in this diagram or in this figure the gel permeation or the gel filtration type of chromatography. Here a column is given A, B, C, D. You can see the various type of color used for showing the size particle. The larger molecules in the size, they are being shown in the red color, and the smaller one they are shown in the green color. You can see the porous packaging material, which can be a bead, that is, or a resin which have been swelled, and in that a sample is being loaded. In your first figure, in your A, you can see a sample is being loaded. So there would be a no graph when you are having a graph for particular solvent system or a chromatographic technique. In that A, you are going to see, because there is no elute over there. No elution, because we are only loading the sample. In B, you are going to see the mixture, the red molecules which are large molecules having the large molecular size. They are being eluted from the beads and they are moving towards the elution or the outlet, because they having a space from the beads. They can't be entrapped in that particular beads, so they can move at a very faster rate. In C, you can see the red molecules, they are being eluting from that particular column, and the green part, they are the inside the column. Okay, so here the graph peak would be increased. And the last one, you can see the green molecules which are of your interest, and the molecular size is small, so they are being purified or they are being eluted from the column. So this is the type of the chromatographic technique based on the molecular size, and here the various gel is going to be used, and the system, buffer system is going to be used, that is to maintain a stationary and the mobile phase for the samples. The next figure also same, but only the colors, they are being different. Here the blue and the brown color molecules, they are being shown, the molecule with different sizes. Solvent system is been there. The blue, they are having the large molecular size, they are being moving faster at the faster rate, and the brown one, it is moving at the slower rate. The blue molecules which are having the larger molecular size, they have been eluted first, and then the smaller molecules, they are being eluted from the sample. Okay, here you can see very simplified figure of the gel filtration assembly. In that column, you can see the gel filtration beads and the protein solution. The molecules, they are shown in the size with same color. In the figure number B, the small molecules, they have been entrapped in the beads or in the resins, and the larger molecules, they have been moving from the spaces present between the beads towards the outlet point, and you can purify both, that is the larger molecules and the smaller molecules. So this is the general assembly which is used in the gel filtration technique to purify the enzyme. Enzyme purification or the protein purification based on the size is generally represented or done in this type of the chromatography, which is the column chromatography. Now we are going to see which factors can affect the resolution in gel filtration chromatography. There would be some factors which can affect your gel, or which can affect your stationary or the mobile phase or your column. So which are these factors which can affect your technique? So first, there are numerous factors which may include the column dimension. The column dimension for this is very important part. So if the column dimension is not regular type, it can affect your chromatography technique, because it required a more time for the resolution or the more time for the separation, and it would be again a costly type of technique. The selection of media, the selection of gel which you are using, composition of buffer, sample volume and the concentration, sample loading and the flow rate. So and last one, it is the length of the tubing. So these are some factors which can affect your resolution in the gel filtration technique. and last but not the least, that is the fraction size. Now we are going to see the factors which can affect this type of technique. Now we are going to say its application. What are the applications for the gel filtration technique? Where we can use this technique and how important this technique can be for the purification of enzymes. So gel filtration technique is a key role for the purification of enzymes like the polysaccharides, enzymes, polysaccharides, that all the biomacromolecules, that is with the enzymes, the polysaccharides, the nucleic acids, the proteins and other molecules. It can be used to facilitate and the refolding of denatured proteins by careful control of changing the buffer conditions. It is used in protein fractionation experiment. It is used in the molecular weight determination of the particular molecules which are in the concentrated solution. It is used for the separation of sugar, proteins, peptides, rubbers and other on the basis of their size. It can be used to determine the quaternary structure of the purified proteins. Now what are its advantages? We have seen its application. What are the advantages of the gel filtration? Gel filtration has the many advantages because it is well suited method and it is very sensitive on the basis of pH, metal ions and the cofactors and the harsh environmental conditions. It has the significant advantage, that is the conditions that can be variant to suit the type of the sample requirement for further purification, analysis and storage without altering the separation. Separation can be performed in the presence of essential ions, that is the cofactors, detergents, urea, guanidine, hydrochloride, which are high or the low molecular ionic strength at 37 degrees Celsius or in the cold room according to the requirements of the experiment. Unlike ion exchange or affinity chromatography, molecules here do not bind to the chromatography because here is we are using the resins or the beads. Okay, so here the mediums of buffer composition does not directly affect the resolution but the degree of separation between the peaks. We have seen a graph in the diagrammatic representation where when the larger molecules they are eluted, the graph peak is very high, and when the smaller molecules they are being purified, the graph peak is very low. So here the resolution would be affected due to the composition of the buffer. It requires a shorter analysis of time. It is well defined type of separation technique, narrow bands, and it have the good sensitivity. There is no loss of the sample. The small amount of mobile phase is required and the flow rate can be set. You can set the flow rate and according to that you can purify your enzymes over here. Now what are the limitations or the disadvantages of this technique? The disadvantages, first advantage, that is the resolution. Okay. The limited number of the peaks that can be resolved within the shorter time scale of the run. So this is the very first important disadvantage of this technique, that is the resolution technique, the peak resolution. If there is shorter period of time, you can't get a resolved type of peak. Filtration must be performed before using the instrument to prevent the dust and other particulates from ruining the columns and interfering with the detectors. The molecular masses of the most of the chains will be too close for the separation to show anything more than broad peaks. If the molecular weight is very close to each other of the two molecules, then it is very difficult by using this technique to plot the graph or to have a peak and to purify them. So it is one type of disadvantage of this technique. So here we have learned about the gel filtration technique. I think all of you have been understand what is the gel filtration technique and how we can perform this. So here we are going to see what we have learned in this session. In this session we have learned the gel filtration chromatography, which is used on the separation of to separate the macromolecule on the basis of their molecular size. Okay, the wide range of gel filtration media which is available and we can choose as per our requirement. We have seen the three types, that is the agarose, the cephaloros. Okay, we have seen that acrylamide gel. So two of them were the natural gels, one it is the synthetic gel. So these types of gel we can choose by our requirement what type of the macromolecules to be purified. Factors that affect the separation and resolution, it may include the column dimension, media, flow rate and etc. Gel filtration is commonly applied for the molecular weight determination. purification of proteins and peptide, desalting and buffer exchanger. So this was the summary. We have seen how the resins should have, what is the buffer here, what should, when we are using any resin or we are using the beads, what should be the nature of that, what gel we can use, what are their chemical properties over there. We have seen its procedure, we have seen its applications, we have seen its advantages and disadvantages on the basis of size. Okay, in our next session we are going to see one purification method which is based on the charge. Every molecule, biomolecule, they are having a charge on them, and on that basis of the charge we can purify that molecule. So here in this we are going to take one example, that is the ion exchange chromatography, where we can use a cation and the anion exchanger. The cation and anion exchanger, they have been used to purify a molecule on the basis of the charge. So in our next session we are going to see its principle, its components, its applications, its advantages and disadvantages. Hope in our today's session you have learned about the gel filtration technique. If you have any questions you can ask me.